branch point A instead of a branch point G. But this one here, if you noticed in the last one and in this one, right, that the intron always has this stuff like this. This one looks like a hand, and the other one had crinkles on the top. Because this processing, this self-splicing is absolutely dependent on the structure of this RNA, of the intron. If you look at RNA in a cell, once again, we talk about RNA, when we think of a linear molecule, we know that DNA forms a duplex, a helix, right? A double helix. But we really don't think of RNA and what it looks like. RNA is never stretched out. We already know it has a lot of protein. But also, it turns out that probably, certainly in the case of ribosomal RNA, 70% of the nucleotides are forming a hydrogen bond with another nucleotide. But rather than being intermolecular hydrogen bonding, in RNA it's intramolecular hydrogen bonding. There's a lot of hairpins and hairpin loops and so forth. And that's what these things indicate. RNA is the bases here, hydrogen bonded with the bases on the other side of the loop. What that does is it gives these RNA molecules a lot of structure. Right? Ribosomal RNAs form they fold by this base pairing, this intramolecular base pairing. And that folding determines where the proteins are arranged on that modified RNA to make a ribosome. So it's important. Anyway, these group 2 splicing, right, what did I say that? They're highly dependent on structure, on the folded structure of the RNA. What that means is if you take this entire primary transcript or that gene and you make a mutation in the intron that changes the folding, then you screw up this process here, right? Now, in spliceosome-directed introns, or introns that are removed by spliceosomes, within the introns there's lots of mutations. You can mutate the crud out of them, as long as you don't change the 5' and the 3' site, and then you still get processing. But in these guys, any mutation in the intron in this region here causes this folding, and if this isn't folded precisely, then that A isn't brought close to there, so that you have that hydrolytic attachment or phosphorylation. Okay, I said it most closely resembles this. Now, if you look at this type of splicing, it only said, did it say 0.1%? It's certainly less than 0.9%, right? If you add things up there. And it's probably less than that. It's pretty rare. And I said it was evolutionarily, it probably was the source of the splicing process itself. And apropos to that, this is very rare, and you find most of it in lower organisms. Most of this occurs in protozoans, right? Why doesn't it occur in bacterial cells? Would bacterial cells have this self-splicing mechanism? No, because bacterial cells really don't have introns. I mean, their goal in life is to keep their DNA at an absolute minimum so that they can divide very, very rapidly. They can replicate that DNA just as quickly as they can, and they can replicate themselves very rapidly. That's their act, that's their mission. Eukaryotic cells are the opposite. Protozoans, of course, are small eukaryotes. They're single-cellular. They're ancient. They're probably the first ones who arose from bacterial cells that had chloroplasts, right, plants. And so they developed this, and this probably evolved in these guys, and it persists. So if you look at some of the protozoans, in a pond today, they have these self-splicing introns. Okay, probably all you ever want to know about self-splicing introns. So here's our gene. We've talked about our DNA. We've talked about our primary transcript. And we've sort of modifying this. We put on a cap, although this doesn't show this transcript with a cap. That really bothers me because that cap is put on right as transcript force on. We've also removed these intervening sequences. But really, the process or the sequence of events is that we cap this guy. Synthesis continues. We put on proteins, HN proteins, as well as these SR proteins. We bind up each of these introns as they come out. We bind up these introns in a spliceosome. And then we put on this 3' cap. And after that 3' cap is put on, or the poly A tail, then this processing occurs and we get the shortening into a message. This is now a mature message that can be transported out. So on the 3' end, we add a tail of between 100 to 250 or so adenosine residues, and it's called the poly A tail, polyadenosine. Many functions of that poly A tail. nucleases have more trouble degrading things. The second thing is that certainly in eukaryotic cells at least, the poly-A-tail is very, very important in protein synthesis. It is involved in the initiation of protein synthesis, and I'll show you that when we talk about it. And then finally, the poly-A-tail is absolutely implicated in the half-life of a messenger RNA, how long the messenger RNA survives. So in the lifetime of this message, when it's first synthesized and it has a couple hundred poly-A residues, as that messenger RNA is in the cytosol and it's being translated, each round of translation shortens that tail. And of course, when the tail gets to a certain shortened length, then the message is turned over or degraded. And there's a definite function on that. We talked about half-life of molecules and stuff, and even of cells. And there's two reasons for that half-life. One is for regulatory reasons, right? The cell needs a particular protein, it synthesizes an RNA, and that RNA starts making protein. But it may not need that protein forever. So there's a determined half-life where it makes that protein and then eventually it's turned over. If the cell still needs that protein, then the gene is transcribed again or continually transcribed. You make more RNA and you keep making the protein. But eventually, when the gene is shut off or the protein's not needed, right, in order to stop the synthesis of that protein, you have to get rid of that messenger RNA. Because if it's there, the ribosomes translate it. So by being degraded like that, it adds half-life to this thing, right? It adds a half-life to that. If you take an RNA molecule, a messenger RNA, or globin, say, and you inject it into a frog egg, an oocyte, a frog oocyte. We use oocytes. Why do we use oocytes? Why would you inject it into a frog oocyte? Why would you do that? Remember, the system to do these experiments is so important. Why frog oocytes? Because you've seen frog oocytes, right? They lay them, they're so big when they lay them, you can look in the pond and see them. So you can inject it. You can put a message in there and say what's going to happen. But could you inject a red blood cell? You can't see it. Possibly you can under a microscope. The easiest thing is to take the biggest cell and inject it, right? So you can take this message and put it into a frog egg. If it's a globin message, if you have no poly-A-tail, that frog egg will translate that message a couple times and make some globin, and then the message will degrade. But if you add a poly-A-tail on it, extend it, then you put it in the oocyte, it'll translate for a week or two, and then it'll degrade. So the poly-A-tail is absolutely essential for the half-life of the messenger RNA. Okay? So how's it put on there? Well, this poly-A-tail is added. There's a site here that's that time is meant to represent. There's a consensus sequence which says, hey, this is the end of the gene, the end of the coding region, right? There's the consensus. This is I'm at the end. You need to stop transcribing here in the transcription of the message and add the poly-A-tail. Okay? That sequence you probably heard. It's called AAUAA. We called it the polyadenylation site. At first, we assumed that the A's were just added on. It turns out now that is really the sequence to cleave the messenger RNA. And this messenger RNA, somewhere between 10 to 30 nucleotides or so, with the 3' sides, so downstream of the transcript, the RNA is cut, cleaved. Right? And that cleavage there, then after the cleavage at this here level, the poly-A-tail is put on, 100 to 250 nucleotides or so. It varies. Now this cleavage is kind of an interesting thing before I go on. Remember, we talked about termination in bacterial cells, and there's row-independent, row-dependent. The messenger RNA, as it's being transcribed, is in duplex with the DNA. And as long as that duplex is there, transcription goes on, the replication mechanism, or the transcription complex continues on to transcribe or copy that DNA. And what happens is, as soon as that RNA comes off, the transcription ends. Well, in this case, there's actually a cleavage event. So to terminate transcription, you essentially need this site here. So this is the poly-A addition site, but it's also the transcription termination site. Because after that, 20 or 30 nucleotides or so is cleaved. Interestingly, right, if you look at this, it says, here's the cleavage, and here's the G-rich region here. It says it's degraded in the nucleus, so this is turned. What's degraded? Well, there's actually a cleavage here. Interestingly, for some reason, and nobody really knows the answer. Once cleavage occurs and you have the end of your RNA molecule, right, the 3' end, transcription continues. That complex, that RNA polymerase complex, the transcription complex, continues going up the gene and making the RNA. Sometimes it puts together 100 nucleotides, sometimes it puts together 1,000 nucleotides. Eventually, because it's GU rich, the whole process slows down, right? It's a very GU rich region of the polymerase complex has trouble going through and continuing to wind, continually slows down and it falls off. And then, that piece of RNA where there's 100 nucleotides or 1,000 nucleotides has to be degraded, it has to be turned into a nucleus. So it's a weird type of a system to end transcription because you still are using energy and nucleotides to make a piece of RNA that has to turn over. So how that evolved, or what the function of that, we really don't know. Okay? But after the cleavage, then the poly A tail is added on. So how come this happens, right? How come this is the way that we terminate messenger RNA, pol polymerase II transcripts, but not pol 1 and pol 3 transcripts? And the reason, once again, is that CTD tail. So riding along on that CTD tail, there are a couple of other proteins that are involved in the processing of the cleavage after that AAU-AAA site. One is called the CPSF, the cleavage and polyadenylation specificity factor, F factors. Cleavage and polyadenylation specificity factor. And it essentially recognizes that AAU sequence. Here it is, right? There's that green guy. It's recognizing this. That's why it has specificity for the cleavage. The other one is called CSTF, which is the cleavage stimulating factor. It stimulates the cleavage. It's not actually the cleaver, but it stimulates it. You can see it's in duplex kind of, or it forms a dimer with CPSF, and that's how it gets to the site. The CPSF recognizes this, and then this guy there stimulates. He's not the cleaver, he stimulates it. There are other proteins involved. So once they recognize and bind to this, there are other cleavage factors, other proteins involved in the cleavage that come into the complex. And then what happens is as they come in, they build this complex, and somewhere 20 to 30 nucleotides or so downstream, 3' they cleave that RNA. Once the RNA is cleaved, then the next step is this poly A polymerase, PAP, poly A polymerase. It comes in and it has that poly A tail. This is one of my least favorite names for an enzyme that people have named in biochemistry. Poly A polymerase. Can anybody think of why I might not like that name? All the polymerases we've talked about already so far, RNA polymerases, DNA polymerases, reverse transcriptases, what do they all need? What do they all require? All require nucleotides, right? Deoxynucleotide triphosphates. What else do they require? Whether it's a DNA polymerase or an RNA polymerase. Did someone say it?
Primer?
A template. Right? They have to have a template. So all the polymerases that you've talked about, if you think about it, right, they have a template that they can read and they make a complementary copy. That's the definition of polymerase. This poly A polymerase, right, there's a cleavage now. All you have is a 3' hydroxyl on the end of a piece of RNA molecule, and it builds this poly A. Right? So to me, this molecule should be called a poly A synthase. It's synthesizing a poly A tail without a template. So really, to determine a polymerase, I don't think so. But anyway, it is called poly. No one listens to me. It's still called poly A polymerase. As that poly A polymerase makes that long poly A tail, right, there are proteins in the nucleus that are called poly A binding proteins, PABP. And so the poly A binding proteins start to bind onto that poly A tail that's being made. Here's a picture of it. So here's the poly A tail, right? There's the CPSF that recognizes AAU-AAA. The cleavage stimulating factor